Okay, Shaita, shall I start? I don't mind if one person is there.
Yes, sir. Pratima is joining in a minute.
Okay, great. Okay, so let me start. So in the last class, I think we have stopped in this part. You all had seen this particular graph which contains the peaks. So now we will go ahead with the analysis of this. So now we will try to analyze the peaks. Already I have told you what are peaks. The peaks are the genome enriched regions, and in the for the chipset experiment, I think my voice is, yeah. For the chipstick experiment, your main target is to identify and then analyze the peaks. Okay. So already I given you for this the explanation for this particular part. Chipstick experiments are designed to isolate the regions and enlist in a factor of interest. Okay, so you have to read it like factor of interest, that is the genome of interest actually. The identification of the enriched region, that is the genome enriched region, often referred to as the peak finding, is an area of research by itself, and that is why chip is mainly used. Okay, and among all the NGS experiments, chip identification is very much common actually. Okay, there are many algorithms and tools for peak finding. You know, already I had seen in the two or three classes before. The choice of this method is strongly motivated by the kind of factor analyzed. Okay, so other things you can read it on your own. It is given quite nicely. I have given to you. So now we will go ahead with the identification of the peaks. Okay, it will hardly take a minute. You can execute it. I hope it is executed for everybody. It will hardly take a minute. So next, what we are doing is, so first we are importing the bed file. Bed file is a binary embedded file. I will explain once I complete this part. So how you can create the bed files. Okay. And now first for identification and for the analysis of the peaks. So we have to the peak is to simply display them in the browser along with the chipsec and the input track. So we are using the annotation track for this. I think it is executed for everybody. So you can see this all the genome enriched regions. Okay. In the last class, we had just done this. What we had done, we had plotted for the reference sequence. So here you can see reference sequence, and here it is for our own sequence. You can check it out here. Refsec is here, and our data are here. Okay. So now you are actually we are getting the particular peaks for our own experiment. So for our own data. So these are all the peaks. Here it is. You can see the maximum, and what are these? We will be checking in the subsequent course. Why you all are joining so late here? All forgot about today's class. Okay. So these are all the, as I told, we have the one of the major function we are using is the MAC. So MSES. So that we are using. These are all the steps for the MAX. So you can see that MAX has successfully identified region showing the high H3K 27 AC signals. We say that both the biological replicates agree as well. However, in some cases, peaks are called only in one sample. Okay, let us go further. So we'll find out the overlaps. So I hope everybody has got this results. Can anybody? I have hidden this particular output. Can somebody share the screen? Can somebody share the screen? Pratima, Hamsini, Saeda, Pratima, can you share the screen?
Yes,
Hamsini,
sir. Yes, sir.
yeah. So you are finding the overlaps at each and every particular step. Okay. So the query length means whatever is your overlaps, you are finding it as 3032. It is given in the last. Okay. So let us, you can unshare your screen. Let us move further. So try to install this package. It will hardly take some time. So again, this Venn diagram is a part of this one, CRAN. So Venn diagram is a very common package. So please try to install that. It will not take much time. If it is installed, please let me know. So what is the area the particular genome is taking is displayed in this particular Venn diagram. So for the REPN1 and REP2, this is the region which is common. Did everybody get it, or still the last command is running? The Venn diagram for everybody, is it the package is loaded? Saida, is it done for you?
No, sir, not yet.
Pratima, Osuri?
Yes, sir.
Okay, great. Osuri? Okay. So Saida, please let me know if it is done.
Yes, sir, it's done.
Okay, great. So we will focus only on the peaks identified in the both the replicate. So in the Venn diagram, the intersection portion is taken for the further studies. So you can see here, already I have written here, the enriched regions are colored in green. So enriched, this is the enriched region, this part, it is colored in green for everybody. And whatever is the present in the particular, it is given from the, you can cross check with the reference sequence. Okay.
Sir, can you share the code to draw the Venn diagram?
Sure. It is already shared.
No, I think you skipped that part.
Which part did I skip? Hmm. This Venn diagram I did not share. Okay, so
No,
yeah,
sir.
sorry. This the Venn diagram is this one. Okay. So Venn diagram, and then for this particular one. So now we'll go ahead with the analysis. So isolation of the promoter. One moment. Okay. Isolation of the promoters overlapping the H3K27 AC peaks. So these are the ensemble from the ensemble data database. I hope everybody knows this ensemble database. From where there we are I trying to isolate the promoters. Okay. So these are the from the Chrome. These are the from the ensembled region data. So it is given here. We will move further. So what we are trying to find out, we are trying to identify the transcription start site. Okay. So we are considering the regions in and around 200 base pairs around the TSS as the promoters. So one thing you can check it out in your data that the strand information is no further there. So next, so we are finding the overlapping promoters. Okay. So how many of the promoters overlap with this particular enriched region? So you are seeing in the data as that you have 364 of the 1973 promoters are overlapped. Okay. So we are calculating how many chromosome or how much part of the chromosome 6 is the part of the promoter region, because you all know that we are the our region identified is chromosome 6. So this is the promoter region which is into which is in the which is our question part, or the region of significant enrichment. So how much fraction it is here? So we are not now finding out the binomial test to confirm the particular region. So probability of success is okay, but there is 95% confidence interval. P value is less than 2.2. Yeah, it is also significant. Okay, we share this code. Yeah. So promoters over with the peak. So you are now finding everything with relation to your enriched region. Already I told you have got the peak. So peak getting the peak is the most important part of the experiment. Okay. So you can check it out that you have already got the promoters, which are the one with the gene ID BRPF1, OGG1, and CAMK1. Okay, already you have got it. So same thing I have given here. The first three promoters identified, it is given there. Then now we move on to the analysis of the distribution of the H3K27AC around the subset of the gene promoters. the way to do. So next we count how many reads are mapping to each type. The resulting vector H3K27AC next is used to create the matrix. Okay, and where each row is a H3K AC enriched promoter. Each column corresponds to the corresponding 100 BP tile of the 2000 BP region, because we are initially, if you remember, we had selected the 2000 BP region. Okay, around the TSS overlapping the H3K27 AC peak. Okay, so let us do this particular part. So finally, we are plotting the result as a heat map and as a plot of the average values for each title for the included promoters. Bigger margins are too large. Bigger margins are too large. Okay, one moment. Let's try to reduce the margin, otherwise the code is all correct. Give me a moment. Okay. So you have got the particular heat map. Screen share properly. Yes. Okay. So you can see the number of reads, distance from the TSS, and the others in the base pair. So you have the mean, you have the promoters, and you have the distance. Okay, I hope everybody has got this. I hope everybody has got this.
Sir, there's an error.
I shared the different code. I deleted and I
Yeah,
shared
even this one is showing an error.
is even this one.
Yeah.
Did you get the results?
No, it's showing an
Pratima?
error. It's showing an error.
Yeah, we will share your screen. Let me see. One moment. Did you miss something? Just come down.
I
Sorry,
think
go
you
up.
haven't
Just
shared the code to make the matrix.
huh?
You haven't shared the code to make the matrix.
I did not share it. I shared it. No. Or did I miss something? One moment. Let me check. If I missed something. You should have to run this together. One moment. I think that is the error. Okay, I missed something. One moment. This is my mistake. Yeah. Now run it. Now run this. I think that was my mistake. I missed some part of the code. Now execute Pratima. Still error. Yeah, you have to actually that first line was missing. First line is missing. So that was the error. So remove everything except the hashtag. Don't remove the hashtag because that is the which is for you to understand. Or you can delete this. No, I think yeah, it is. Remove this code and execute again. Delete it from the site. There is the option in the top. You can delete the code. In your right hand side, hmm, now execute the full code. Let me see. Still error is coming.
Sir, the code above this where you made the matrix, you didn't send that.
See the error is coming. Error in age, error in evaluating argument X in selected method image not found. Did you miss any part of the code? Is everybody getting the same error? Error image. Are you using from your mobile, Pratima?
Sir, before this, we made a matrix, right?
Sorry?
We made a matrix before this code, right? You haven't shared the code for that.
Ah,
You only shared the introduction.
is it show? One moment. Yes, okay, let me check it out if I missed something. Mm, okay, I think this part I did not share. Oh, okay, I got it. This part I did not share. That is why it is coming. This H327, this is just run this one and then try to do this. Hopefully it will work. Hmm. Yes. Yeah. I somehow I missed that code. Yeah. You got it correct now.
Yeah, thank you, sir.
Yeah. Okay. So this is I concluded this part. Now what I will do in the next class, I will let you know to help you out to prepare the data set. Okay. That we have to do it in Linux. That is the part. Because we have to import the data, because as I told you, I have used the data from some other sources. Now I have to let you know so that you will be able to create this particular data and you can work on your own. So all these things, because already I told the data is given to you, they just extracted the data. So that is not the part, but already you have executed. So now you have to generate your own data and you have to go ahead. Okay. So that part we will be doing from the next class. So today I will be ending the class here. Every others, those who have joined late, and if you have not done it, please execute all the codes. And maybe tomorrow I will come and I will be same time, that is 6:30 tomorrow. We will start with the Linux codes. Okay, is it fine with everybody? Please do not be late. I know today is Sunday, but since in the initially I was my health was little not fine because the weather here is not good, so I could not take one or two classes in the initial days of this week. So no doubts, tomorrow, if you are comfortable, we start by 6:30. Okay. Okay,
Yes,
then
sir.
see you. Good night. Hmm.